Hello, my name is Makoto Arita, team leader of Laboratory for Metabolomics, RIKEN IMS. Today, I'm going to talk about cutting-edge lipidomics technology reveals the biology of lipid diversity and disease control. So this slide shows the emerging diversity of lipids, in other words, structural diversity of lipids in nature. As you can see here, lipids are categorized to 8 categories that includes fatty acids, glycerophospholipids, sphingolipids, and sterols, and so on. And if you consider about the head group difference and also acyl chain diversity and their combinations, the total number of lipid diversity is roughly calculated to be more than 100,000 species present in nature. So the question is how those lipid diversities are regulated and also generated, and what happens if those balances are dysregulated, especially in human diseases. So to address this point, we developed LC-MS-MS-based advanced lipidomics platform. As you can see here, there are different types of mass specs, including triple quad MS and also QTOF type MS. And these, mass spec as a combination, so we can address deeply about the structural diversity of lipids. By using triple quad MS, we can do the targeted lipidomics analysis that is highly sensitive and quantitative method. That is ideal method for measuring lipid mediators or bile acid or so on. In addition, we actually do the non-targeted lipidomics analysis by using QTOF type MS that can cover wide range of lipids and semi-quantitative analysis that covers phospholipids, sphingolipids, and sterols and so on. So this method is biased method, and also we can cover more than 8,000 molecular species of lipids. So in year 2020, we published a seminal paper describing a platform on non-targeted lipidomics. As you can see here, from biological samples, different types of lipids are extracted and analyzed by the QTOF type MS, and after that, computational analysis and molecular networking is performed, and then now we can analyze more than 8,000 lipid structures unbiasedly. The key technology for this method is a software called MS-Dial4. As you can see here, based on the LC retention time and also collision cross-section from ion mobility separation, and also parent MS and MS-MS fragment, now you can narrow down and annotate the structure of lipids, candidate lipids. And even though you don't know the structure, still we can do the de novo analysis by calculating the composition formula and also provide the ranking like this. And this software can be downloaded from RIKEN website under this URL. So as a result, now we start to appreciate that there are more complexity and also structural diversity of lipids present than we originally expected. And some of them are quite new structures, and some of them are present uniquely in certain tissues or organs, and also some of them are present in certain group of cells. And this kind of MS-MS spectral library or information was achieved by measuring more than 1,000 biological samples from a range of sources, including human blood, mouse tissues, other mammalian cells, microbiota, algae, and plants. So this is a kind of blue ocean in front of us. But the problem is that lipidomics by itself is made from homogenate of tissues or cells and that was the spatial information. So to cover this point, now we are trying to develop a new technology, including spatial lipidomics together with spatial transcriptomics and proteomics to capture the local distribution of lipids and their spatially heterogeneous metabolic dynamics. And this is so-called lipidome atlas, which we are developing now. And by doing that, we aim to elucidate the mechanisms that create, regulate, and recognize lipid diversity and its localization in vivo, and to elucidate diseases caused by the disruption of such mechanisms. So this slide shows the spatial lipidomics to create a lipidome atlas. The advantage of the method is ion mobility separation utilizing collision cross-section value of lipids, and also MALDI2 post-ionization to reduce ionization separation. So by utilizing different values of collision cross-section that can be predictable by machine learning, so now we try to develop the method to visualize like thousands of different lipid species from single specimen or single cells. So this slide shows a workflow of MALDI imaging mass spectrometry. The first, you need to prepare frozen tissue sample as a section, and then spray matrix and irradiate a laser to get the imaging and data analysis. And after that, remove the matrix and perform the post-staining, like HE staining or the antibody-based immunostaining. And by doing that, so you can address which lipids are localized to which cell type of which layer of tissues by using the same tissue section. So this is an example of spatial lipidomics of retinal tissue. So as you can see here, you can see retinal layers and also a cornea in here. So we monitored more than hundreds of different types of lipids as a positive ion mode, and some of them are selectively localized in a retinal layer, and others are localized in different places. And this is negative ion mode. Again, you can see hundreds of lipids, different types of lipids, and some of them are localized in a retinal layer, and the other are in other places. So this type of new technology will enable us to address a question like how DHA-containing lipids are enriched in the retina. Acyl-CoA synthase 6, ACSL6, is an enzyme which is important for enriching DHA in the retina. As you can see here, di-DHA-containing phosphatidylcholine, PC226226, is uniquely present in the retina, and here is a volcano plot comparing ACL6 knockout mice and heterozygote, and there are a group of lipids highly reduced in knockout mice, and PC226226 is included here. And if we look at the tissue localization of this unique lipid, you can see the yellow signals in here, and that is specifically localized in the retinal photoreceptor layer, and that is totally abolished or absent in ACL6 knockout mice. And this type of technology and information will enable us to further address the mechanisms that define the localization and distribution of characteristic lipids in the layered structure of the retina, and also address the causes of retinal dysfunction associated with aberrant lipid metabolism in ACSL6 deficient mice. This innovative lipidomics technology will lead to the creation of the lipidome atlas that will facilitate the discovery of new biological systems shaped by lipid diversity, leading to an integrated understanding of lipid-centric biology. Thank you for your attention.